The process of breaking down fats into smaller molecules is called emulsification. If you were to pour water and vegetable oil into a jar together, close the jar and then shake it, you'd see that the two would mix for a while. What you've done here is emulsify the oil. You've broken down the big molecules into small molecules by shaking, and those small molecules then are more easily surrounded by the water for a time. Eventually the water and the oil separates again until physically emulsified again. Bile chemically emulsifies fats in the duodenum. So with fats that have been emulsified, that is broken down into smaller molecules, and carbohydrates and proteins that have been broken down into small polymers, the hydrolases of the pancreatic juice can now go to work to break down these molecules into monomers and other very small molecules that can be absorbed into the body. Let's look very specifically at the enzymes that are secreted by the pancreas to break down the molecules of your food. Remember that there are 4 classes of biological molecules. 3 of those are abundant in food and used by your body. Those are carbohydrates, proteins, and lipids. Before we move on, though, to talk about how enzymes work on these big molecules, let's remind ourselves of some terms. Carbohydrates and proteins are polymers made up of monomers. Monomers are small molecules, and when multiple monomers are linked together, we call the resulting big molecule a polymer. Mono means single, poly means many. Can you remember what the monomer subunits are of carbohydrates and proteins? Let's find out. So this is going to be clicker question number 4. And the question is asking about carbohydrates. Most carbohydrates are polymers made up of what kind of monomer? This is a multiple choice question. Go ahead and take a minute to choose your answer, enter it, and store it into your clicker. Continuing with carbohydrates as an example, let's review how monomers together make polymers. Remember that generally, polymers are made up of monomers. So carbohydrates, proteins, and nucleic acids are all polymers made up of monomers. We often are interested, though, in looking more closely at these biological molecules, and when we do, we want to know the specific monomer unit that makes up the specific polymer. So in carbohydrates, as an example, the monomer unit is the monosaccharide. Saccharide refers to sugar. Mono means single, so a single sugar. Monosaccharides combine to make up polysaccharides. A polysaccharide could be a polymer of 2 or more polymers. If it's 2, we call it a disaccharide, as you see here. If it's 3 or more, we call it a polysaccharide. As an example, the body encounters the monosaccharides glucose and fructose that you see here. Glucose and fructose might bond together to make a disaccharide, which is sucrose, like this. And many glucose monomers may be linked together to make big polysaccharides like starch, which is a storage form of glucose in plants, or like glycogen, which is the storage form of glucose in animals. Okay, so now that we've reviewed some terminology, let's go back to the enzymes that are at work in the pancreas. Name some of these enzymes and look at what they do specifically. So in the duodenum, the hydrolysis of carbohydrates is facilitated by the enzyme pancreatic amylase. If you remember, amylases are a group of enzymes in general that break down carbohydrates. So pancreatic amylase is the enzyme secreted by the pancreas. Pancreatic amylase is specific to breaking down starches. Remember that starch is the plant storage form of glucose. So pancreatic amylase breaks down starch into its glucose monomers. The enzymes sucrase and lactase, which are secreted by the pancreas, facilitate the breaking down of disaccharides, sucrose and lactose, into their monosaccharide units. Proteins are broken down also by the hydrolysis reaction and facilitated by general classes of enzymes called proteases and peptidases. These facilitate the breakdown of proteins into smaller polymers, into peptides, and into amino acids. Remember, amino acids are the monomer unit of proteins. The last class of biological molecule that is broken down in the duodenum under the action of enzymes secreted by the pancreas are lipids. The pancreas secretes a general category of enzymes called lipases. Lipases break down fats that have been emulsified first by bile. Let's review where the different biological molecules that make up your food begin and end chemical digestion in the digestive system. Let's put this chemical digestion into the context of a piece of cheese pizza. Cheese pizza is made up of crust and tomato sauce, which are both mostly carbohydrates, and cheese, which is made up of mostly fats and proteins, although cheese has some carbohydrates in it too, like lactose. Chemical digestion will begin in the mouth, but just of carbohydrates of the crust and in the sauce. This digestion will occur by hydrolysis, facilitated by salivary amylase. Can you remember the accessory organs that secrete salivary amylase? Proteins in the cheese won't begin to be digested until they hit the stomach. Here, pepsin will be used to break down proteins. In the duodenum, carbohydrate digestion will finish by the action of hydrolases such as pancreatic amylase and lactase, and protein digestion will finish with hydrolases such as proteases and peptidases. The fats from the cheese and oils of the pizza won't begin to be digested until they reach the duodenum after they have been emulsified by bile, so they are more susceptible to the enzymes secreted by the pancreas, which are lipases. The action of bile, this emulsification, is an analogous process to mechanical digestion by chewing. Emulsification reduces big fats into small molecules that then have a lot more surface area that are susceptible to lipases. Chewing a piece of pizza into smaller bits increases the surface area so that enzymes have greater access to the molecules of the food. So at this point, we've talked about how you've ingested food and how your ingested food has been digested. So even though you've ingested food, you may think the food is in your body, but it really hasn't yet entered into your body. To be in your body, the products of digestion need to cross the epithelial barrier and enter your cells. So the monomers that result after the digestion of your food have to be absorbed. This occurs in the small intestine. The interior of the small intestine is designed specifically for this purpose. What you see here is a cross-section of the lining of the small intestine. What you should notice first is how the lining is folded. And then looking at those folds, notice that the folds have their own extensions that are called villi. Villi in turn have their own extensions, which are called microvilli. This cross-section of intestinal tissue shows the finger-like projections that are the villi. Extending from each of those projections are even more projections that look like loops of thread. These are the microvilli. The tissue that makes up the microvilli is made up of a layer of cells that are designed to absorb nutrients. The design of the small intestine, the folded tissue with villi and microvilli, creates a lot of surface area to pack in as many of these cells as possible. The surface area of the small intestine is maximized relative to the volume that the small intestine occupies in the abdominal cavity. In fact, if you were to take all of the tissue of your small intestine and lay it out flat on a surface, it would cover as much area as the size of a tennis court. So we take all of that tissue and fold it into folds upon folds upon folds to make it fit into the abdominal cavity of the human body. The other detail to note about the structure of the small intestine is how blood vessels course through the tissue that makes up this organ. Nutrients are absorbed across the lining of the small intestine, and then enter the circulatory system so that they can be carried away and delivered to other tissues and cells of the body. After nutrients have been absorbed from the chyme by cells of the small intestine, what's left passes through the large intestine. The large intestine is certainly wider than the small intestine, and shorter too. By the time the chyme reaches the large intestine, it's made up largely of indigestible food matter, such as cellulose from plant tissue, and water. The job of the large intestine is primarily to absorb water, but also small ions that are still part of the chyme. Also, the large intestine is home to a diversity of bacteria that are mostly harmless. Here, the bacteria live off of undigested materials of the chyme. The beneficial byproduct of the bacterial activity are vitamins, such as vitamin K and B12. So another function of the large intestine is also to take up vitamins that are produced here. A non-beneficial byproduct of bacterial activity is that they make gas too, which is then the cause of flatulence. What's left over makes up the waste product of food processing, feces. One other structure of the intestines that's of interest to us is the appendix. That's this small finger-like structure here at the junction between the small and large intestine. For a long time, we didn't know what the function was of the appendix. We only knew that it could become infected and require surgery to remove it. And because many people live a healthy, normal life without an appendix, it was thought to have served a function that had once been lost. In the last few years, though, biologists have revised this hypothesis regarding the function of the appendix. It's now suggested that the appendix is home to a variety of beneficial bacteria that are there to renew beneficial bacterial populations in the body when needed. For instance, remember those bacteria that live in the large intestine and make vitamins. Well, if you get sick and you take antibiotics to combat a bad bacterial infection, you'll kill a lot of the good bacteria in your body as well as the bad bacteria, because antibiotics aren't usually specific to just bad bacteria. So the good bacteria that are protected in the appendix are ready and waiting to recolonize your gut when needed. In both the small and large intestine, the intestinal wall can become inflamed as a result of what are known as autoimmune diseases. These are diseases in which the immune system of the body turns against itself. Celiac disease has been mentioned before in lecture. This is where cells of the immune system are triggered by wheat gluten to break down the joints between cells of the small intestine. This causes the leakage of nutrients in the small intestine so that they're not taken up, and also causes a wide range of other symptoms. Crohn's disease is another autoimmune disease that causes inflammation along the lining of the entire intestinal tract. Symptoms of Crohn's disease include diarrhea, vomiting, weight loss, and abdominal pain. It's a very serious disease, thought to be genetic in origin, but one that we don't know much about at this point. The last 6 inches or so of the large intestine is the rectum. This is where feces are stored until the last step of food processing, which is elimination. The anus is the second opening of the alimentary canal, which controls elimination. So that concludes our tour through the digestive system, where we've looked at the structures of the alimentary canal, the accessory organs, and the functions of all these organs together. We've also talked about the steps of food processing, and let's have our last clicker question for the lecture, which tests how well you understand the steps of food processing and how they differ. This is a multiple choice question, clicker question number 5. Go ahead and enter your answer into your clicker and store it there. The steps of food processing provide the body with the raw materials needed to maintain homeostasis and proper function of the body's organ systems. But not all food is equal in providing for the body. The body obtains 3 necessities from food: fuel in order to do cellular work, the building materials for making new molecules that keep cells and the body functioning, and essential nutrients that are required for new molecules, but also for keeping the body running smoothly. We'll talk about these necessities in the next lecture.